Okay, we go now to the citric acid cycle, or known as Krebs cycle or tricarboxylic acid cycle. The Krebs cycle is also called citric acid cycle. It is the fundamental metabolic pathway involving 8 enzymes, which is essential for the energy productions through aerobic respirations, meaning the energy productions of the Krebs cycle involves the use of oxygen because it is considered to be aerobic respiration. And like glycolysis arose in early in evolutions because the product of this one, the oxaloacetate class, is also a building blocks, okay, in the production, of glucose under gluconeogenesis. That is why the pathway is very, very important of biosynthetic building blocks like oxaloacetate, which is also a product in the product of gluconeogenesis, which is found in the cytosol, because pyruvate is converted to oxaloacetate, then oxaloacetate to malate, and malate back to oxaloacetate. So oxaloacetate is one of the product also of citric acid cycle. So amino acids is the biosynthesis and fatty acid biosynthesis. These are the important source of biosynthetic building blocks of the gluconeogenesis, amino acids, and fatty acid synthesis, which is known to be the oxaloacetate, the product of the citric acid cycle. The citric acid cycle occurs inside the mitochondria in the process described, and then we have the reducing agent, the nicotinamide adenine dinucleotide plus hydrogen, and the flavin adenine dinucleotide plus 2 hydrogen. They are responsible for producing the ATP in the electron transport chain. Okay? So electron transport chain, ACA or ATC, is the process which involves the NADH and the FADH2 produced during glycolysis. So we have a beta oxidation. Other catabolic process are oxidized, thus releasing energy in the form of ATP. Meaning, the NADH and the or nicotinamide adenine dinucleotide plus yung FADH2, known as flavin adenine dinucleotide, they are involved in the transport of the electrons that happens during the process of glycolysis. Okay, and the utilization or the use of that reducing agents will release energy in the form of ATP. Okay, so step 1, pyruvate and coenzyme. Okay, we have here the pyruvate, which is the product of the glycolysis process. Remember, so combine with coenzyme. So combine to form acetyl coenzyme A. Okay, by enzyme pyruvate dehydrogenase, or known as pyruvate dehydrogenase enzyme. The acetyl coenzyme A, a 2 carbon atom, combines with oxaloacetate, which is also the product, of the gluconeogenesis. Remember, okay? Combined with oxaloacetate, okay? Combined with oxaloacetate plus yung acetyl coenzyme A, so the product, or it will form a 6 carbon compound known as citrate, which is catalyzed by enzyme citrate synthase enzyme. So that is a hydration process, okay, because water is added. Okay, take note, yung step 1, yung pyruvate combined with coenzyme A, acted by enzyme pyruvate dehydrogenase. Of course, this is a release of carboxyl group. At the same time, NAD is converted to NADH plus hydrogen, so that is considered to be dehydrogenation process. And there is a reducing agent known as NAD. So the product of pyruvate and coenzyme combination is acetyl coenzyme A. So acetyl coenzyme A combines with oxaloacetate, which is also the product of citric acid cycle, and also an intermediate product of a gluconeogenesis process. So after the combination, so it will form a product known as citrate, acted by enzyme citrate synthase. So this is known to be the hydration process. Hydration meaning you add water to form citrate. Okay? So step 1, this is 1, oxaloacetate plus acetyl coenzyme A. So acted by enzyme citrate synthase. So the product is citrate. Next, we have step number 2. Yung citrate class is isomerized to isocitrate. Take note. From citrate is isomerized to isocitrate to eliminations of one molecule of water. Okay? So there is a elimination of water. But before magkaroon ng formation ng natawag natin na isocitrate, first yung citrate, there is what we call dehydration process that is acted by enzyme aconitase. So yung citrate will form cis-aconitate that is acted also by aconitase enzyme. Then another reactions, no, in that step number 2, this is the intermediate product. So cis-aconitate undergoes hydration product. Okay, that is what we call this one, addition again of water to form dextroisocitrate. So this is the intermediate product, yung elimination of water kanina. So take note citrate to cis-aconitate. So there is removal of water. So it is acted by aconitase enzyme. At the same time, water again is added plus yung enzyme aconitase. So the product is dextroisocitrate. Step number 3 class, the isocitrate, okay, is oxidized and reduced by NAD, that is converted to NADH plus H, that is dehydrogenation process. And then the two recently acquired carbon from acetyl coenzyme A are oxidized to carboxyl group. So the resulting compound is alpha ketoglutarate or alpha ketogluttaric acid. Isa parehas lang yan class ang alpha ketoglutarate at the same time yung alpha ketogluttaric acid is almost the same. So in step number 3, remember class, yung isocitrate, so it will be oxidized and reduced, no, by the reducing agent NAD that is converted to NADH plus H. Okay, and then it undergoes dehydrogenation process that is acted by dehydrogenase enzyme. So there is a removal of carboxyl group. It will form alpha ketogluttaric acid or ginatawag natin alpha ketoglutarate. Okay, so makita natin yung legend, oxygen, hydrogen, sulfur. So yung NADH is nicotinamide adenine dinucleotide. We have your pyruvate dehydrogenase enzyme, adenosine triphosphate, guanosine triphosphate ito. Okay, so that is for step number 3. Okay, next. So step number 2, try to say, i-structure natin, we have citrate. Okay, so magkaroon na yung natawag natin na rehydrations or dehydration, I mean. So it will form a cis-aconitate. This is known to be the intermediate product. Okay, removal of water. Then at the same time, magkaroon ng addition of water, that is rehydration. It will form isocitrate. Okay, so kung i-reverse natin yung reaction class with aconitase, so there is dehydration. It will form cis-aconitate. At the same time, mag-add ka ng water, it will form again citrate. Okay, so this is considered to be yung steps wherein citrate act as a base. Next, step number 3, yung isocitrate plus NAD. So ang product is alpha ketoglutarate or alpha ketogluttaric acid. It's almost the same ng class. Okay, so step number 4, okay, yung alpha ketoglutarate or alpha ketogluttaric acid, take note, is further oxidized by another NAD plus converts to NADH, that is oxidation process. The last carbon remaining from pyruvic acid is removed. So there is what we call removal of the carbon dioxide. The resulting molecule is then attached to the coenzyme by a bond and not stable to form succinyl coenzyme A. Okay, that is step number 4. Okay, so this is step number 4. So alpha ketoglutarate is oxidized to form succinyl coenzyme A. Okay. So step number 5. So yung coenzyme A is displaced in the phosphate group. So yung GDP is converted to GTP. So this phosphate group is taken by guanosine diphosphate to form guanosine triphosphate, a molecule with similar functions to ATP, meaning ito yung source ng energy. So GTP is used to generate ATP via substrate level of phosphorylation process, wherein yung succinyl coenzyme A class magkaroon na tawag natin na phosphorylation process, and the product is known to be succinate that is acted by enzyme succinyl coenzyme A.